Livestock, livestock products and microbes. A story of animal, human and environmental health. Livestock-associated microbes and their interplay with the host animal are crucial in animal health, particularly during challenging life events such as weaning and parturition, while environmental health is threatened through their role in emissions of greenhouse gases and nutrients. Further down the consumption cycle, intestinal microbes interact with human health when digesting animal products. These are some of the research topics addressed by the Laboratory for Animal Nutrition and Animal Product Quality, which consists of 3 research units. We are pleased to briefly introduce the research focus of each of these 3 units. Enjoy! Given the role ruminants can play to convert low-quality feed into milk and meat of high nutritional value, the mission of our group is to advance knowledge on ruminant nutrition and microbial digestion towards a resource-sustainable animal nutrition, responsible food production, and healthy productive animals. As such, we are targeting several of the Sustainable Development Goals. To limit the number of experimental animals required to study microbial digestion, we rely on well-designed in vitro simulation tools, which are complementary to animal studies. These simulations aim at representing specific digestive processes and are customized according to the research question. As such, these simulations can be used, for instance, to assess the nutritional value of novel feeds, to screen the effectiveness of detoxifiers such as mycotoxin binders or degraders, to evaluate the effectiveness of rumen bypass technologies, or to test feeds and additives for their potential to reduce nitrogen or methane losses towards the environment. Moreover, this technology can be used to gain mechanistic insights in the kinetic of these digestive processes, as well as in the growth, community composition and activity of rumen microbes, and this both in the short term as well as over a period of up to 14 days. Obviously, in vitro results should be validated in vivo, with particular interest to monitor physiological changes of the dairy cow and her microbiome during the challenging transition period around calving. Our main expertise within this in vivo approach relates to the use of milk metabolites, in particularly milk fatty acids, to mirror the rumen microbial metabolism in relation to the cow's physiology. These milk biomarkers are currently used to complement external data and information gained from other milk diagnostics and sensor data. Both our fundamental as well as applied research topics and methodological tools fit within the European key research areas related to animal production systems, particularly in research and innovation tackling resource use and implementing circular economy principles at animal level. Furthermore, in the livestock sector, the relation between animal nutrition and health is another key area for further investigation. The interaction between the host and its microbes has been identified as one of the major missing links in this respect. Finally, given the importance of enteric methane emissions by ruminants, sustainable reduction of these emissions is key within the European Green Deal target of becoming the first climate-neutral continent.
Many people love their piece of meat. However, producing quality pork or chicken meat entails growing these animals in a healthy environment and feeding them well-balanced diets. Animal feed does not only determine 50 to 70% of the production costs, it also highly impacts the sustainability of the production system and the well-being of our animals. In our research, we develop expertise in monogastric animals with particular focus on disadvantaged production stages, such as the weaned piglet or the heat-stressed broiler chicken. The mission is to increase our understanding of the feed-gut interaction in order to improve nutrient use efficiency, animal performances and gut health. We focus our efforts on 5 key topics in monogastric nutrition and management. Already on the first day, there's potential to support our animals. For example, we study how we can improve the intake of colostrum and hence increase the survival of newborns. We also execute nutritional research at this early stage and, for example, study how probiotics and prebiotics can improve the maturation of the gut. This has shown to be helpful to limit the chance for digestive problems later in life. In this regard, the link between animal behavior and nutrition also gets our attention. As you might know from human babies, learning how to eat and accepting new feed sources demands social learning. We execute research to find the optimal way to boost social competences of our pigs. For example, having families interacting with each other can improve the resilience of young animals. In this field, our activity is not limited to research. We also try to provide management guidelines and aim to increase the adoption of new nursing strategies on the farm. Paramount to our research is the development of adequate tools and methods. Therefore, we apply various in vitro and ex vivo models next to our models or facilities for raising pigs and broilers. We employ these methods and infrastructure to evaluate novel feed ingredients such as insect protein, microbial protein and fermented protein sources. These novel feed ingredients may contribute to a higher sustainability of the production chain. However, in most cases, they are too expensive for common feed formulas. Therefore, our focus is to establish their added value to promote gut health. For example, the black soldier fly larvae contains high amounts of lauric acid that can act as an antimicrobial. Over the last decades, our group has been researching many supplements that can support gut health, with particular focus on essential oils, medium-chain fatty acids, antioxidants and functional amino acids. For example, our research on essential oils has taken a turn. Before, we focused on antimicrobial properties. Now we focus on the interaction between specific channels on enteroendocrine cells in the digestive system that mediate their action on gut motility and the secretion of digestive fluids. Continuous improvements in animal nutrition are key. However, we also believe that the digital revolution can create a better future for production animals. In our research group, we employ camera technology and electronic feeder stations to monitor feeding behavior of weaned pigs. This is because the amount of feed an animal consumes is useful data to tailor diets to the needs of individual animals. We also use these data to make predictions on the onset of digestive diseases. We foresee a future where real-time monitoring tools and big data analysis support farmers in making decisions for the benefit of the animal. Finally, our broiler research is focused on the effects of heat stress on the physiology of the bird, and in particular on the oxidative status, the cecal microbiome, and more recently on bone development. These findings aid in the implementation of new nutritional mitigation strategies. The changing climate and the reallocation of broiler production to subtropical regions urges us to foster this research. Our method development work and research topics well align with several European key research areas in animal production. The study of the microbiome is core in most of our studies, and we intensify our efforts for implementing digital technologies in research and on-farm applications.
Meat production and consumption is heavily scrutinized nowadays because of animal welfare concerns, for its environmental impact and for the link with several chronic diseases. Yet, meat plays an important role in society and in the diet of many people. And the mission of our group is therefore to advance knowledge on the quality of meat derived from sustainable production systems and its role in human diets, thereby contributing to achieving several of the Sustainable Development Goals. In response to the consumer trend for eating less but better meat, there is a renewed interest in Europe in the use of local, more rustic breeds from more extensive production systems. As an example, we contributed to research on the historically renowned Menapian pig. Current research focuses on how the eating quality of meat from several species is linked to the production system from farm to fork. Our toolbox of sensory and technological meat quality measurements and our knowledge of animal production systems allows to assess which factors, genetic factors, nutritional factors, are determinant for high quality meat. Similarly, we are interested in the value of meat as a source of high quality protein, long-chain polyunsaturated fatty acids, minerals and vitamins, and unique compounds such as carnosine and anserine, histidine-containing dipeptides that display several physiological roles. We found that the contents of these dipeptides strongly differ according to the breed of chickens. The analysis of fatty acids in feeds and in different animal source foods, meat, milk and eggs, is a key expertise and workflow at Lanipro. Our previous work on omega-3 fatty acids in meat has contributed to the establishment of several meat labels.
Despite its nutritional value, a high consumption of red meat and particularly processed meat is associated with an increased risk to develop various chronic diseases in observational population studies. However, the explaining mechanisms are not known yet. Our research aims to elucidate how different meats and processing techniques, but also interactions with other food items, influence the formation of various toxic metabolites during gastrointestinal digestion, such as products from lipids and protein oxidation and protein fermentation. For this purpose, we use a static in vitro digestion model in which we expose meat to saliva, gastric juice, duodenal juice and bile. By using this approach, we found, for example, that phenolic-rich spices and herbs are very efficient inhibitors of oxidative reactions during digestion of meat. Meat consumption may affect health through many different pathways, for example by influencing oxidative stress, the large intestinal microbial community and inflammation. We study these pathways in animal feeding studies using rats and pigs. Hereby, we not only give attention to the intrinsic meat characteristics and meat processing techniques, but also to dietary interactions with other food items. In addition, hybrid meat products and meat replacers more and more mimic the taste and appearance of real meat products. However, its impact on nutrition and health is largely unknown and will be investigated in our future research.
In summary, our research on the various qualities of meat related to the production systems fits into the farm-to-fork strategy as part of the European Green Deal, whereas our research on the digestion of meat and its plant-based alternatives fits into the Food 2030 research priorities, moving towards more sustainable food systems with increased quality standards for the production of safe, nutritious and tasty food.